impulses. So basically we have this mechanism of myelinating our neurons, and myelin is a bunch of proteins and phospholipids that lay down this protective sheath that helps to insulate the movement of ions down the axon. Okay, and so myelin is present along the length of the axon, not continuously. It's actually present in intervals, right? And so just to remind ourselves of the direction of the stimulus, the movement of the stimulus. So it's perceived here in the soma, and then this is the general direction, right? It must proceed in that direction. And so we start at the proximal end of the neuron, and we move the depolarization events. So these are intervals of the membrane that are becoming depolarized at a time. Okay, and so we don't depolarize the entire length. That's how this neuron, that's how this action potential is being propagated. And that's why we say that we fire an action potential, because it sort of rips down the length of the axon, all right, from proximal to distal. So just to loosely describe this here, we have one segment of the membrane becoming depolarized. This segment of the membrane will be in its refractory period. This segment of the membrane will be at rest, right? And then we have signals or influence from this depolarization onto the nearby cells, such that the impulse moves from one segment to the other, down the length of the axon. Okay, so they go all the way down the length of the axon. They happen at every patch of the membrane, so different segments or intervals of the membrane are sort of truncated, and they depolarize independently of the next segment, And the conduction rate is slow, typically because we have action potentials generated at each spot. So each spot has to generate an action potential, then the next, then the next, then the next. This is where myelination comes in. This is how myelination speeds up this process. Because we have myelin at one of these segments, or covering a span of the neuron, of the axon, we actually jump over that myelin as we conduct this impulse. So in that way, myelinating myelinating the nerve or the neuron is really facilitating the conduction, right? The faster conduction of this action potential. We'll point out here at the end that the action potential conducted has the same amplitude. We don't lose the signal the way we did with a graded potential. So it would be tempting to think that because it's moving further away from the original site of stimulus, that we lose some of the intensity, and we don't, right? We spoke about properties of the action potential that ensure that it is self-regenerating, and it is going to be very same to the next. Okay, so to speak about this in a sequence here, we have a site in the membrane, right? That's depolarizing. We have a site that's at rest. This part of the membrane becomes excited, and you can see that by the change in the charge, right? The change in the charge, such that exciting the neuron makes it more positive inside as that sodium rushes into the cell. The next site, site B, was at rest, but now it's been influenced by site A, such that it now becomes depolarized. So site B is now repolarized, site A is in its refractory period, site C is at rest, okay? And then the next segment becomes depolarized. So site C now gets influence from these voltage-gated sodium channels, right? And let's go back a little bit to the mechanism that we talked about earlier. So because one voltage-gated sodium channel is influencing its neighbor, right? This is how the next segment becomes depolarized, and not the previous segment, right? The previous segment is in its refractory period, and so we physically cannot fire another action potential there. And so that prevents the action potential from moving in the retrograde direction. It must proceed in one direction. Okay, and let's look at what myelin does for this process. So myelin is present in intervals, again, along the length of the neuron. It's not continuous. Some neurons are more myelinated than others. Some are unmyelinated. And again, that will vary depending on where in the body that neuron is, what the conduction velocity is that's required for that specific function or location. And so the action potential is jumping along these nodes. So the spaces in between the myelination occur every 1 to 2 millimeters, and that is called a node of Ranvier. This is a heavy area, or a heavy concentration of voltage-gated sodium channels, right? And that makes sense because that's the area that's really being depolarized. and we're going from one node to the other to the other. So the action potential is leaping, and so we describe it as being saltatory. Saltatory from the Latin word saltere, which means to jump or to leap, and that's exactly what it's doing along the nodes here, the unmyelinated bits. Okay, so again, just highlighting that very same thing. This is keeping the action potential in one direction. The polarization is happening in this direction. The repolarization is happening in that direction. So as we depolarize this segment of the membrane, this segment of the membrane becomes repolarized, et cetera, et cetera. Okay, and just to compare here, difference in myelination. So again, we heavily myelinate neurons that are responsible for faster conduction velocities. Think about skeletal muscle, think about glands, think about peripheral nerves. Those are heavily myelinated. Longer, bigger nerves, sciatic, radial, those types of nerves have a much heavier myelination, and that is to speed up their conduction velocity. And that makes sense. They are longer, they span more of a length in terms of from their soma down to their axon terminal. And so having a faster conduction velocity in these sites makes sense. Whereas other neurons of the autonomic nervous system, of cardiac muscle, are not as heavily myelinated. And that makes sense. We don't need faster conduction velocities for those functions. Those are unconscious functions most of the time, blood pressure regulation, things that we're not actively trying to control. And so we don't need faster conduction velocities for these types of fibers. Right, and so a very thin, unmyelinated neuron may conduct at a speed of 1 meter per second, whereas a well-myelinated, thick myelinated neuron will conduct at a speed of 100 meters per second, so much faster in terms of its conduction velocity. And really pointing out the underlying mechanism, and that's because the myelination allows the action potential to jump. So if it's a bigger myelination, it's a bigger jump, right? It's going to move faster along the length. All right, so let's